Hello everyone, today I'll introduce clone multiplex IC kit for you. I'll introduce from the following aspects. What can we offer, why to choose us, and where to use multiplex IC kit. First, what can we offer? Clone multiplex IC kit, which is based on fluorescence immunoassay, can be used to detect multiple targets at the same time. In addition to multiplex IC kit, we also have more than 7,000 FLIA kits for single analysis involving more than 10 species and covering more than 2,000 targets. The multiplex IC kit uses magnetic microspheres as the carrier to detect the targets by double antibody sandwich method or competitive inhibition method. The core of this technology mainly includes 2 points. First, magnetic microspheres have been color-coded into 100 spectrally distinct regions. Each of these microsphere regions can be quickly distinguished by the Luminex analyzer. Second, the specific antibodies for different targets are coupled with microspheres labeled with different fluorescence. Thus, the different targets in the same sample can be quantitatively measured simultaneously. This slide shows the test principle. Take the multiplex IC kit that use double antibody sandwich method, for example, or premixed magnetic beads coated with antibody captures the target in the standard sample, and then binds to the built-in labeled detection antibody. The surface of the magnetic beads forms an antibody-antigen antibody sandwich structure. Then the complex binds to the streptavidin-coupled PE. For the reading, the magnetic microspheres are suspended in the sheath fluid, passed through the injection needle and entered the Luminex 200 analyzer. As shown in the figure, the red laser emission signal identifies the magnetic bead region and classifies the magnetic bead to determine the target being measured, while the green laser emission signal identifies the PE fluorescence intensity for quantitative measurement of the target. Here list part of the customized panels, such as cytokine panels, chemokine panels, metabolism, hormones, etc. This slide shows the standard curve of an example of the multiplex IC kits. At present, clone multiplex IC kit can simultaneously detect 8 targets in one run. The 11 targets are divided into an 8-plex panel and a 3-plex panel. Let's show you the original data of the multiplex IC kit. This is the display of original data of 8-plex cytokine panel. This is the display of original data of 6-plex cardiovascular disease panel that obtained with multiplex IC kit. The detection instrument we are using now is Luminex 200 analyzer. The analysis volume is 70 microliter. The beads used are magplex microspheres, and the instrument is set to collect 50 microspheres for each target in one sample and generate median MFI value. As you know, microsphere count of 50 is enough to produce a statistically accurate result. The Luminex 200 analyzer is calibrated and verified weekly, using the corresponding calibration kit and the verification kit. This ensures the accuracy of the test and data. In addition to the popular panels, we can professional to develop your own multiplex panels according to the experimental needs. This slide shows a customized flow of multiplex IC kits. Firstly, communicate with the customer to gather the needs, then analyze the targets. The next step is to develop the multiplex IC kit. As soon as the kit is successfully developed, It will be delivered to the customers if they want to make the experiment by themselves. We can also provide experimental service and data analysis service if the customers don't have an instrument. Second part, why choose us? What are the advantages of clone-clone multiplex IC kit when compared with ELISA kit? First, multiplex IC kit can be used to detect multiple targets at the same time. One sample can test 8 targets simultaneously. Up to 100 targets can be extended, and it only needs 20 to 100 microliter of the sample. This saves both time and money. Second, the raw materials which are produced by ourselves is 100% traceability. More than 7,000 FLIA kits for single analysis involving more than 10 species and covering more than 2,000 targets can freely match customers' experimental needs. Third, the sensitivity of multiplex IC kit is 10 to 100 times higher than common ELISA kits. Fourth, the detection range can be expanded to 1,000 to 10,000 times than the same type of ELISA kit. And the last but not the least, the results of a multiplex IC kit are more stable, accurate, and reproducible with at least 50 magnetic beads read per for each target. We promise 100% quality guarantee. Our products meet the quality management system and have rigorous R&D processes and strict 3-level quality control, including raw materials, semi-finished products, and the final products. We also promise 100% service guarantee, provide technical support before and after sale, and promise promptly reply within 24 hours. The third part, where to use multiplex IC kit. Multiplex IC kit can be used to make quantitative analysis for various biological samples, including not only common samples such as serum, plasma, cell lysis, and tissue homogeneous, but also some unusual samples such as saliva, tears, cerebrospinal fluid, etc. Multiplex IC kit can be used in a variety of different research fields such as tumor research, drug research, immunological research, and so on. More and more researchers choose this new detection technology in their research. This research is working on organ transplantation. We can see clone-clone multiplex IC kit of cytokine and chemokine used in this article. So does this article. Clone-clone multiplex IC kit of immune response was applied to the field of cardiovascular research. There are also articles published using Luminex kits, which are same-type products as clone-clone multiplex IC kit. They allow researchers to qualify multiple biomarkers simultaneously. We can see multiplex kits are applied to many different research fields involving infection, inflammation, stem cell therapy, metabolism, tumor therapy, immunotherapy, and other fields. In summary, clone-clone can provide multiplex IC kits involving more than 10 species and more than 2,000 targets, together with experimental service and customized service. Second, clone-clone multiplex IC kit can be used to qualify multiple targets with one sample at the same time. Besides, the kit has many advantages, such as higher sensitivity, wider detection range, 100% traceable raw materials, more stable results, and 100% quality and service guarantee. Third, with clone-clone multiplex IC kit, customers can make quantitative analysis for various sample types, conduct the research in different scientific fields, and publish high-level scientific papers. Thank you very much for your listening.